Okay, I want to begin by acknowledging that the land on which we gather is the unceded traditional territories of the Coast Salish peoples, including the territories of the Musqueam, Squamish and Tsleil-Waututh nations. And second of all, to welcome you here to the Vancouver Aquarium, an ocean-wide initiative. So I'm going to play you a short video about Oceanwise, in case some of you have not been here before or don't know too much about it.
I'm a friend of the ocean.
I am an interpreter of the ocean.
I'm a really big fan of the ocean.
I am a child of the ocean.
I am a geek of the ocean. I am a voice for the ocean.
I'm constantly amazed by the ocean.
I am deeply moved by the ocean. I am mesmerized by the ocean.
I am
The ocean.
I am a defender of the ocean.
I am guardian of the ocean.
I'm a diehard supporter of the ocean.
I'm a fighter for the ocean.
I'm truly appreciative of the ocean every moment of every day.
I'm an advocate for all the animals in the ocean.
I'm truly grateful for the ocean.
But I think hopefully you maybe got the message. Now our mission here at Oceanwise is to inspire both the local and global communities to become ocean-wise. Our overriding mission is to increase the understanding, wonder and appreciation of our oceans, empowering all of us to help protect our ocean health and our planet as a whole. So yeah, first of all, thank you. Yeah, second of all, thank you so much for coming tonight. And also, yeah, would like to thank Science Literacy Week. So the reason that we put this awesome event on and give you some delicious food is because of Science Literacy Week. We got a grant from this organisation to run this event today. And I don't know if you've noticed, but this week, I think from the 17th of September to the end of the week is Canada's Science Literacy Week. So if you haven't noticed, there's Science Literacy Week events all across the city. UBC have put on some at the Vancouver Public Library, for example, so keep an eye open for other events coming up in the last few days. So yeah, I would like to give a huge thank you to this organisation for being able to fund this event. And also thank you for coming out in the terrible rain. I hope the food and a beer made up for it. And the great talks, hopefully. So the talks that are coming up, first of all, I'm going to give you a quick overview about, I've called it From Science to Science Communication. As this is Science Literacy Week and we've been funded by that organisation, I thought I'd give a quick overview as to why I think that is such an important thing, especially in today's climate. And then Katrina Wilson will be talking about taking the aquarium to the people. So she'll be talking about her mobile aquavan that goes out to communities all across the country and allows people who might not otherwise have access to be able to interact with invertebrates and so on that we take for granted here because we're so close to the coast. And then Eric Solomon will be talking about another of Oceanwise's projects, outreach to Inuit youth. We have and then a couple of research talks. So Oceanwise does a lot of research on a variety of topics. So we have one talking about ocean microplastics, and then Brittany will be talking about her life as a marine biologist studying whales. So let me see where we got to. So yeah, I'll get started on my presentation. So yeah, again, I can't say it enough, thanks to Science Literacy Week for allowing us to do this. And yeah, so science communication and science literacy is extremely important to me. So the reason for that is because I've completed my PhD in August 2017. So I did my PhD at UBC, where I was studying the behaviour, ecology and evolution of a particular species of spider, social spiders that lives in Ecuador. And I thoroughly enjoyed my PhD. I learned an immense amount. But during my time working with some fantastic scientists, one thing I did come to realise is the immense importance of science communication, of reaching out to the public and to communicating everything that's going on, all the really important, and sometimes, not so important, just exciting and fun science that goes on. And so because of that, because I realised just how vital this was, in 2014 to 2017, I became executive coordinator of Let's Talk Science UBC. So this is a science outreach organisation that sends students to community events and classrooms across BC to get young people enthusiastic about science. So because I enjoyed that so much and because I found it so rewarding and such an important thing to be doing, when I completed my PhD, I decided rather than continuing into an academic career, I would continue instead to spend my time promoting science literacy, doing science communication, and trying to reach and teach as many people as possible exactly what science is, why it's so important, and why it's extremely fun. So why is it so important? So we obviously all know the vague reasons, but I just like to repeat them so that we kind of all remember and can appreciate just why this is so vital. So obviously, in today's climate, we have quite a few issues. In the socio-political situation that's happening right now, it's becoming more and more obvious that a science literate population and science communication, effective science communication, is vital. So, for example, 58% of Canadians lack a basic level of science literacy. And this is a figure that I found, it's from 2014, and this is actually pretty good worldwide. Like, Canada's pretty good on this, but it's still shocking that so many Canadians lack basic science literacy. And science literacy also goes along with critical thinking. So I'll give you hopefully a very brief example about, a fun thing that happened, one that doesn't have too many consequences, but an example of how this can manifest itself. So I did a lot of my fieldwork for my PhD in Ecuador, because that's where the spiders that I studied lived. And obviously Ecuador is on the equator, so you can go to a big monument on the equator, and you can get tour guides to do a demonstration of the Coriolis effect. So the Coriolis effect is when the water on one side of the equator rotates one way when it goes down a drain, but the opposite direction when it's on the other side of the equator. So these guides will step 1 meter into the southern hemisphere, pour some water in, and it swirls round one way, and then they'll step another meter to the northern hemisphere, pour the water in, and it swirls the other way. So obviously, having a little bit of critical thinking and science background, I was like, that doesn't make any sense, because how can 2 meters make such a difference? And so I asked the guide, and he said to me completely openly, oh yeah, I just pour the water in, and I swish it one way, one side with a bucket, and a slight wrist movement the other side does it the other way. And I was like, yeah, fine. I mean, it's just a bit of fun. You get a couple of dollars, and it's a bit of fun. But you'd be surprised that even the people that he was talking to didn't believe him, even though he said this. And then I told a lot of people, I got the tour guide to admit that it is just a little bit of a trick. So many people did not believe me and swore blind that they saw the water rotating one way and rotating the other way. So that's just, it doesn't really matter. It's not going to affect anybody's lives whether they believe that or not. But it's just a little bit of an example where sometimes a little bit of scientific knowledge and a lack of critical thinking can bring you to the wrong conclusion. And so also that can also be problematic with the media. And it's not just, I was going to say today's media, but this has gone on for a long period of time. You've all seen these headlines, these scientific headlines that they don't really make a lot of sense. A lot of times they're reported wrongly, or the context isn't reported correctly, and it can be very confusing and overwhelming if you don't know really how to make your way around it. So I'll give you a little bit of a quick example. So I was just doing a Google search of, to be honest, on the Mail, because I thought they might have some slightly dodgy science, and I found this one. So fracking causes as much seismic activity as jumping off a ladder. So controversial method for extracting gas is extremely unlikely to trigger an earthquake we would feel. So you can see down here, the date is April 2013. And then I found this article sorry, it's slightly mixed up. So this is from July 2013. So a couple of months later, they had the headline, "Fracking could lead to an earthquake domino effect." So if you're reading this, how do you know, unless you do a lot of research yourself, how are you to know what to believe, what to think, and what to make basically of the world? So also another reason why it's important to be science literate and to use scientific evidence when deciding what course of action to take, effective policy, is that sometimes things can be counterintuitive. So this is a quick example of a study that was carried out in 2005. And so it's carried out specifically on this leaflet here, which was published by the CDC. So everyone's seen these, they're myth, fact posters or leaflets that people distribute on a wide variety of subjects. So this one by the CDC was about the flu vaccine. A very legitimate aim is to try to get more individuals to be vaccinated. However, when scientists studied what the effect of this was, they actually found, disturbingly, that people that read this leaflet actually misremembered the myths as fact. So they forgot about the facts and just, for example, thought that a flu shot couldn't cause the flu. This effect got worse over time. So over time, people were more likely to believe the myth and completely forgot about the fact. And they even attributed this information to the CDC. So it's had the completely opposite effect to the one that was intended. So unless you actually studied this, I don't think you would have thought that would be the case. So anyway, in conclusion, if you're ever going to do any communication, never ever use this facts myth, because it does, not only does it not work, it actually makes the situation worse. And then another small example of how things can turn out as you would not expect is a study done in 2000. So they were looking at ways in which to encourage parents to come to daycare to pick up their children on time. So one of the things, a logical thing that they did, was that they decided to fine parents if they arrived late. So if you arrive late, you've got to pay $5, for example. So they thought this would solve the problem. In fact, the complete opposite happened. When they fined parents were much more likely to turn up late to collect their children. And they think this is because by monetizing it, they thought, well, parents would think that, oh, they're late, but that's fine, I'll pay the $5 and that will make up for it. So in fact, by charging parents, again, it made the situation worse. So these are just some examples, and these counterintuitive situations happen in a wide variety of cases. So it's important to be critical thinking and to study things and listen to what the science tells you. And then obviously there's some issues with science and literacy. So obviously we have, the old climate change problem. So there's a big issue with people understanding exactly, what's happening, why climate change is happening, what the difference is between climate and weather. Vaccines. This is a situation that I think, it does appear to me to be kind of getting worse. We have measles outbreaks kind of all over the place now. And this is obviously very problematic because people are no longer, only a small number of people need to abstain from vaccinating, and it can cause a big problem. And then GMOs could potentially solve a lot of problems, but because they're misunderstood and I would say, to some extent demonized, that we're not using the technology to do all the good that it could potentially do. And then also, people in general are really bad at analysing risks of situations. So this is another situation where a little bit of science literacy could go a long way to help people. So yeah, so this would also cause problems. So, you know, in the health arena, it can be extremely confusing. You've seen in the also the headlines all about health issues. How can you, you know, chocolate makes you happy, chocolate makes you sad, coffee extends your life, coffee causes you to die early. It can be extremely hard to navigate all of this. And so the science literacy can have a real problem if people partake in you know, in things that aren't scientifically proven. It can cause problems. So, for example, there's a study I think that came out in the last few months that suggested that people on the Atkins diet, for example, actually have a shortened life expectancy because they're not getting sufficient variety of foods. So that's one example. To being an engaged citizen and to be able to hold our elected representatives to account, it's important that you understand and can critically analyse what's communicated to you and what politicians do and stand for. And then again, climate change. This is a big issue, and it's a multifaceted problem. So being able to understand and not be scared of the science, I think would be fantastic. And if you're science illiterate, then as we can see, this is causing massive problems, and the problems aren't being solved right now. So, and then some fun misconceptions of science. So first of all, I think a lot of people think this is what a scientist is. Somebody in a lab coat with a pipette in a lab. But really, science is a lot like this. So for example, my science, I did all my science in the jungle, running through the Ecuadorian jungle, looking into spiders' nests. I didn't spend any time in a lab. And then also, perceptions of scientists primarily as white men with beards in lab coats. So this is a particularly depressing Google search. I searched scientist cartoon. This is what I got. And not only are they all men, they're also all chemists. I mean, I have nothing wrong with chemistry, but I think quite a lot of scientists are not chemists. So I would like to change it more to this. And this is much more realistic of what science and scientists actually are. So with that, I'm just going to very briefly run through a little bit of what we do here at Oceanwise, and why I'm so happy to work here and to be promoting science literacy and doing science outreach to try to alleviate some of these issues. So, yeah, as I said before, our mission here is to inspire the global community to become Oceanwise. So, Oceanwise itself is kind of the umbrella organisation that we have the Aquarium is an part of Oceanwise. We have Oceanwise Education, which is the department that I work for. And then we have Oceanwise Research, and we have a couple of people in that department who are going to talk to you today about some of the awesome research that we do. So this is a broad organisation, and then I'm just going to talk about education because that's what I'm passionate about. We have all sorts of things where we try to reach out to people all across the board. So we have camps and clubs for young people and mentorship, Katrina's mobile programs, and so on. So for my department, I do primarily adult education, so you're at one of these events. So the next event coming up is on the 25th of September, and it's Can Sustainable Plastics Save Us, so that should be interesting, and there's only a few spots left, so if you're interested in coming along, you could jump onto the website, ocean.org/Ocean Matters, and register for that event. And some other events coming up. So I run a lecture series that happens every 4th Tuesday of the month. So the October lecture, we're talking about sea star wasting disease that's actually happening right here on the BC coast. And then in November, we have a talk about ocean-wise plastic research. So we have a very large department here that does a lot of research on microplastics and plastic pollution, which is a very new and emerging field. And there's so much more to learn and so much that we don't know, that it's fantastic to be having some cutting-edge research happening right here in the building. And then also, I live stream everything. So there's a camera over there, which is currently filming everything that's happening. And so all these lectures go up online. So if you ever can't make it, you can live stream in, or you can watch the video online later on. And then also, my last little plug is I also run a program called Film Forum. So this is an international program trying to get OceanWise's message and the message of ocean conservation to as wider audience as possible, in as many locations as possible. So the way that this works is people apply. So if you have a organisation that would maybe like to do this, you can apply online to host a film forum, and then in this case you'll get to choose from one of these two movies. We supply the movie. We've already paid for the fees for screening the movies, and then arrange for an expert or two to speak either in person or remotely after the movie. So we have one happening in Mount Vernon in a couple of weeks. There's one that's being ran at Langara in, again, a couple of weeks. So if you're interested in perhaps hosting one of these film forums, either speak to me afterwards, or you can go online and apply to host one of these. So again, thank you so much for coming. As you can tell, I'm extremely passionate about science literacy and science communication, so thank you again, Science Literacy Week. And thank you for partaking, for coming along. And so this event was completely covered by the Science Literacy Week grant, but normally the events that we put on are not funded so lavishly. So, for example, I try to make all the events free and to make them as accessible as possible for as many people as possible. So, with that in mind, there's a couple of donation boxes at the back if you fancy giving us a loonie or two to allow us to run more programs in the future. That would be absolutely fantastic. So, with that in mind, I'll have maybe a couple of minutes for questions if anyone has them, and then we'll move on to the next talk. So, does anyone have any questions? Okay. As we're running a bit late, that's probably not a bad thing. Right, so the next talk is Katrina Wilson. So she's manager of mobile programs here, and she'll be telling you all about the adventures of the Aquavan. Okay. Do you know where your talk presentation is? Yep.
Good evening, everybody. Thanks for your patience. I noticed there's a lot of people standing at the back, and there's a few chairs up here, so if anyone wants to come forward and share a seat, I'm sure people wouldn't mind you coming to share the table with them. I am the manager of mobile programs here at OceanWise, and Ruth stole a couple of my slides, so I'll be really, really quick. She did mention we are now OceanWise Conservation Association. That is the overarching organization under which we all live: aquariums, education, research, and direct action. So for me, in education, all of our programs depend on all you scientists out there discovering things and giving us the information so that we can take it and go out to schools or run camps or whatever it is that we're doing in the education department and tell people about it. For my team, this is a list of all the parts of education that we have here with a picture of my beautiful aquavan, we have children and youth. They are the birthday parties, sleepovers, camps, clubs, curriculum programs, the kinds of programs that schools book to come here on a field trip. Service learning, which sounds really dull, but is actually lots of fun. It's things like Great Canadian Shoreline Cleanup, empowering people to go out and actually make a difference in their own communities, wherever we are. Public programs, you all know what that is. And Ocean Bridge is another new program for us, involving 40 youth from across the country, from coast to coast to coast. And if you want to learn more about that, I'm sure there's more information on the website. And then, of course, the best one, mobile programs. So how do we use science in mobile programs? We use it in lots of different ways. My team has a very diverse background. Most of us have degrees from universities of one sort or another, most of them in science, but not everybody. Some people have experience from life experience, doing different things that has brought them to the same place. So my team is amazing. I love my team, and they are all very good at what they do. And they take what you do, scientists. Hands up, scientists out there? Lots of people? Excellent. We take the great work that you guys do, and we help the general public. That's what Tamara's doing in this photograph here. We have a mobile aquarium. We have that out. People can touch and feel the animals, learn a little bit about the animals, about the ecosystems that they live in, and why people living in Manitoba should care about the oceans, because we're all connected. In other ways, our experience with several versions of the Aquavan, we've come up with new and maybe better ways to do things. And as I mentioned, we incorporate all of the new science stories into our programming. We have a lot of different ways that we do this with children. Most of what we do is go to schools across the province, across the country, and we run programs teaching children about the ocean, but also trying to make them think a little bit more critically. We do this through a few programs. One of them is a program that's all about the scientific method. We have a station where we put out artifacts, and the teacher has to run the station, kids with her students, using the scientific method as the base for asking questions, trying to get the kids to come up with what they think these things might be for. They may be a mystery skull that we don't even know what it is. Then we run a program that we call Conservation Research in Action. Now that is a program for secondary students, and we talk a lot about different programs that are running here at Oceanwise Conservation Association. We talk about the House Sound Research Group a lot, using sponge reefs. We talk about sea otters, urchin, and kelp, because all of our programs use our live invertebrates as sort of the hook to get people interested in what we're talking about in the first place. Then we start sliding all the other information in there too. So one of my favorite programs is Art of Experimental Design, which sounds like a terrible program, but it's where we actually have the kids running experiments with our animals. We take some animals into schools, and the kids start the session without touching anything. They're just using their observations. I'm just going to put my glasses on so I can read some of the observations. It looks like a Johnny Test sign. I don't know what that is. It looks like a cactus made out of snail eyes. It's a spiky gumball. It looks like a cactus made of hair. Looks like a baby basketball. It looks like a kiwi. And they're talking about a sea urchin, and this is before they've touched it. Then they start to come up with questions. We invite the students to come up to the board, and they all write. They love writing on the board. So we get them to start writing their thoughts, their questions on the board, and there are a lot. I'm going to try and pick out, how does it move? How does it eat? Why is it spiky? Why does it have lots of spikes? There are lots and lots of different questions. And we don't tell them what the animal is. Many of them have never seen. Anyone know what it is that they're looking at? did I? Oh, it's a sea urchin. Yes, sorry. I'm talking so much I didn't even notice. So it's a sea urchin. But they have so many questions, and the program that we run this with, they're usually like grade 4, 5 age. So they're so excited about the process, talking about their own observations, their own questions. And then we have the kids actually design an experiment that they run with our animals under guidance to answer one of these questions. Obviously, there are certain questions that we won't be able to answer in the time allowed. You know, how does it reproduce? We can't do that in the time allowed. But we can talk about it. It's a way just to engage the students. It's a really exciting program for me to run in schools. And here you have students doing a little food choice experiment. They're looking closely at their sea urchin. There's also a hermit crab in the other dish beside them. And we purposefully have the students use, for example, different shaped containers. And then there's a discussion at the end of the program where we talk about, you all did this experiment. We don't encourage all of the groups to do the experiment the same way. They all do it the way they would like to. They all do it slightly differently. And then we talk about whether or not we can actually combine all of the data that we've collected today. Maybe it's which food does a sea urchin like to eat? Maybe it's how fast can it turn over if you flip it upside down. They're very creative with the different kinds of experiments that they can do. And we encourage all that diversity in the classroom, and then we talk about why or why not you can or cannot combine all of that information together. Again, just trying to start them thinking critically about the things that they're doing, that they see in the world. With secondary students, we run programs mostly where we talk about classification, but wherever we can, instead of just the straight taxonomy or why these animals are grouped together, we're also trying to talk about the ecosystem that those animals live in, why it's important that we protect that ecosystem or that animal. Most people don't really care about sea anemones that much. But when you take the whole ecosystem, people are much more inclined to care about it. We hook them in by using the live animal. And this is almost the end of my talk. So the impact that we have just with mobile programs, I'm giving you big numbers here. In 2017, we spoke with over 63,000 students. We had over 91,000 conversations in different communities across Canada. So our total was over 154,000 conversations, connections with people in different communities across the country, which is really important to me for science literacy. As Ruth has mentioned, it's really, really important to get as many people as possible caring for our oceans and taking action in their own communities. That's the call to action for us in our programs, is that we are hoping to encourage all of these kids, all of these participants, to take direct action in their own communities. And how we do that is by taking the great information that you guys give us and using it in our programs. People want to know what is happening out there in the world. They want to know what scientists are saying about kelp forests or whatever it might be. And we can be part of the conduit to those people. That's really all I have to say. Any questions? Thanks.
So our next talk, if he's about, I've just spotted him, is Eric Solomon. So Eric is the director of Arctic programs here at Oceanwise. And he's dedicated much of the last decade working with Arctic indigenous communities and scientific researchers. So he'll tell us about some of his experiences and the extremely awesome and important program that he spearheads. So I think it's this one. Is this one? Is this me? We've called it the same, which is sensible, I suppose. No? This one. Oh, there it is.
good evening. Thanks very much for coming out tonight. I don't know what the weather is doing now. It's supposed to be pretty rainy, but I appreciate you all coming tonight. I'm going to talk about research, but I'm going to talk about a kind of a hybrid of research and engagement of youth and communities in the North. We do have a lot of different Arctic research programs, ranging from studying beluga acoustics and communication to microplastics in the Arctic, to biodiversity of the subtidal in various locations around the Arctic archipelago. But I'm going to focus on one specific program called ECARVIC and talk a little bit about that program and some of the research that we're doing with Inuit youth in that program. I'm going to start by, see if I can do that, there we go. I'm going to talk just very briefly about change, and I'm not going to throw up any graphs and lots of statistics at you because, quite frankly, I don't want you leaving crying. But what I will do is just point out a couple things just to make sure you have the idea that things are changing in the North, and they're changing very rapidly. The Arctic of today is very different than the Arctic of 10 or 15 years ago even. We're losing about a little more than 13% of our Arctic sea ice at the end of summer every decade. We are looking at about a 40% loss of sea ice compared to the 1979 to 2000 average. A lot of different changes are occurring, and I just want to make sure that everybody's aware of that and recognizes that there are a lot of changes, and we'll talk about some of the impacts of those changes in a bit, but I didn't want to dwell on that specifically. I'm also not going to talk a lot about different Arctic species. This isn't a talk about that, but there is one in particular that I do want to talk about. One species that lives in the Arctic that is greatly affected by all these changes that are going on with sea ice and other things as well, and that we don't really talk enough about here in the South. So I will skip that and go right to that. It's this species. You might recognize it, right? People live in the Arctic, and there are 100,000 or so Canadians who live in the Arctic. 85% of them are Inuit. They're indigenous people. They live in communities that range from 500 to 1,800 people, some more than that, some less than that. This is, on the left is Darlene and her son Brian and her nephew, and in the middle is another friend of mine that I've known for quite some time, and Randy. They live in Pond Inlet, and Pond Inlet is the community that you see there on the right. Way up in the high Arctic, it's an isolated community. They rely greatly on the sea ice as a way to travel, as a platform to travel on, as their highway, as a platform for hunting. In this case, they rely quite a lot on hunting and harvesting from the land in order to support their families. So these people are greatly affected by the changes that are going on in the North. There's a lot of research that does occur, and I'll back up to that for a moment. There's a lot of research that does occur, and it needs to be happening. We're really in a race to try and understand the changes that are going on and the impacts of those changes. But at the moment, what we understand is far more limited than what we don't understand about these changes. That research that's going on is incredibly important, but a lot of it, in fact, most of it, almost all of it, the research agendas are set in the South for the North. The questions that are being determined are being determined in the South. The research methodologies are being determined in the South, and the research is happening in the North. Inuit communities are not necessarily engaged in that research and engaged in setting those research agendas the way they could be. At the same time, there's a very strong and continuous call for more meaningful engagement of Indigenous people and communities in research and the integration of Indigenous knowledge into research and science, but across Canada. So whether that's the Nunavut Land Claims Agreement from 1999 that really established Nunavut as a territory, the UN Declaration on the Rights of Indigenous Peoples or the TRC calls to action, there is a strong call for more engagement of Indigenous people and more integration of Indigenous knowledge into research. At the same time, these communities are very young. So 60% of Nunavut is under 30. Youth are not sitting around waiting to inherit the world around them. They're increasingly being asked to take leadership roles and to play a strong role within their communities. So about 9 years or so ago, when we were starting to think about what our organization should and could be doing with regards to research in the Arctic and with regards to engagement of Indigenous communities in the Arctic, I was up in Pond Inlet and ran into the person on the left there. That's Shelley Elverum. She lives in Pond. She has for a very long time. And Shelley was working with a number of youth in that community, asking a number of questions about some of the kinds of things that I was just talking about. And we started to talk about what kind of a role our organization could play and what kind of role youth could play in the North in making differences in some of these areas. And together, Shelley and me and this group of youth came up with the idea of Ikarvik. Ikarvik is, the word means bridge in Inuktitut. And Ikarvik is a program that works with Northern youth to be a bridge between research and their community. And what we really try and do is empower those youth and their communities to identify and act on their own research priorities. So come up with what their research priorities are and provide the capacity for them to begin to work towards acting on those research priorities. And at the same time, it's finding ways to build skills and confidence and experience, the kinds of things that young leaders need to be able to take on roles within their community that can make a difference. So what that looks like, I'm not sure if you can read that, but that's okay, I'll explain it. It starts with youth identifying and exploring the strengths of something called Inuit Kayumayatu Kangit. Sorry to our interpreter for that one. It's often called IQ. And IQ is it's Indigenous knowledge, Inuit knowledge, values and customs kind of all packaged into one. So it's not just what we know, but it's how we've come to know it. It's how we engage with that knowledge and with each other and the environment and so on. So they spend time identifying the strengths of IQ as a way of knowing and describing and understanding the world. And then they spend some time thinking about the strengths of science as another way, not necessarily a better way, a different way of understanding and describing and knowing the world around you. And by thinking about the strengths of these two ways of knowing, they can start to think about how these two ways of knowing can work better together to address issues of local concern. And once they've done that, they do start to identify issues that they think are important for their community. So these are often environmental issues, but in the North especially, even though anywhere really, you can't separate the environment from the social and cultural and political and economic contexts in which the environment occurs. So they might say, we're concerned about, where all the char are, but that issue isn't just an environmental issue, that's a food security issue as well. It may be an economic issue. So they're identifying issues that are important for their community that could become research priorities. Once they've done that, they then present their ideas to members of their community, leaders, elders, others in the community, and they come up together with a kind of consensus on a list of research priorities for the community. Now the community has said, okay, these are the things that we think need to be studied, need to be better understood, using both IQ and science. The next step is to connect those communities and the youth with researchers who don't just have the expertise, but also have the right kind of mindset to want to work with youth and want to work with communities as partners to address one or more of those research priorities. So whereas most often the case would be a researcher would come into a community with a set of questions already in mind and be looking for help from folks within the community one way or another. This kind of turns it around, where the community says, these are our questions. The researcher is there to help from the science standpoint, and the community members are there to help from the IQ standpoint, and they put these things together. The youth are a part of that whole process. They are trained as co-researchers in that process. So they're a part of coming up with the research questions. They're a part of determining the methodology. They're a part of collecting data, whether that's interviewing elders and hunters for information or whether that's going out in the field and collecting data other ways. They're an important part of communicating back to the community, as well as the results, communicating the results of the research. They'll come to the Arctic Net Conference, for example, and present to 200 scientists and things like that. So they're a part of that process all the way through. And that's kind of how the program works. And what I want to do is just quickly just show you some of the research projects that youth in their communities have been involved in over the last year or two. So the first one, Pond Inlet. One of the key concerns that the community had come up with was a concern about the potential for invasive species. So these are species that don't belong there, that show up one way or another, that could potentially wreak havoc. And these invasive species can show up because of climate change, or they can show up because they're in ballast water or on the hulls of ships. So the community is really concerned about that because there's a mine not far from there, and there are ships actually coming in to get the iron ore almost every day and releasing ballast water and so on. So the community is very concerned about the potential for invasive species as a result. So the youth then, these Akarvik youth, teamed up with Kim Howland from Fisheries and Oceans, and they learned how to collect seawater samples and process those seawater samples, and then those samples get shipped back to Laval University for more processing. And what they're looking for is environmental DNA. So the idea here is that everything that's crawling around in the ocean or swimming in the ocean is shedding its DNA into the water all the time. And so if you take a sample of that seawater, you can actually see what DNA are floating around in that water, and by identifying that, you can then determine what species have been crawling or swimming around in the water. And so to date, they've identified about 250 different species using environmental DNA. The good news is so far none of those have been determined to be invasive species. But this is the beginning of a monitoring program for the community to be able to watch out for invasive species over time, with the youth conducting the research and communicating that back to the community. In the communities of Saluit and Nunavik, and Joahaven and Pond Inlet in Nunavut, the youth have been involved in a program called Arctic Corridors and Northern Voices. And that program is run out of the University of Ottawa with Jackie Dawson as the lead on that program. And in those communities, as well as several others we've talked to, one of the big concerns that the communities had was increased ship traffic in the area. And you know, as the Arctic becomes easier to access, you get more ship traffic, and we are seeing more ship traffic around these communities and elsewhere in the Arctic. And so there are concerns about how these ships may impact wildlife as well as impact people who are trying to travel across the ice or on the water and so on. And so this project works with the youth to train them to conduct research by interviewing and doing mapping exercises with hunters and elders and others in the community to identify areas that are important for wildlife. So maybe it's where polar bears are denning or seals den or caribou are crossing or that sort of thing, as well as important areas for people. So areas where people are doing their hunting or they're traveling and that sort of thing that could be impacted by ship traffic. And this information then is going to, of course, back to the community, but it's also going to the Canadian Coast Guard and Transport Canada, who have proposed shipping corridors for the Arctic and said, these are areas where we think ships should travel. And so it's going back to them to inform that process with the community's perspectives and ideas and thoughts about ships and ship traffic. So helping to address their concerns and that issue. In Kugluktuk, Nunavut, one of the key priorities, the key concerns was the health of the Coppermine River. The Coppermine River is a hugely important river. It's source of drinking water. It's what people travel on both in the winter and in the summer. It's an important source of food for people, catching char as well as whitefish and so on. And people in Kugluktuk had seen a number of changes over the years on the river in terms of water flow and level and how cloudy the water is, the turbidity, things like that. So working with Heidi Swanson and Rosie Smith from University of Waterloo, the youth developed a monitoring program for the river. They spent the beginning of this summer putting out loggers and sensors and all kinds of things to measure and monitor the river water and get a better understanding of how things were changing over time. And that'll continue for about 3 years or so. So the last one that I wanted to mention is out of Pond Inlet and recently worked in Arctic Bay as well. And these communities, along with most communities, to be honest, have expressed concerns about the changes in sea ice and sea ice becoming thinner, more dangerous to travel on, more difficult to predict, and so on. And so working with Trevor Bell and a program called Smart Ice out of Memorial University, the Ikarvik youth worked with their community in a workshop format to understand what issues the community had, what concerns they had about traveling on the sea ice and the areas they were concerned about and so on. And Smart Ice then developed a program where these youth are going out and mapping and measuring and monitoring sea ice conditions using all kinds of interesting equipment, including a radar type system that looks at how thick the sea ice is in real time, so they can travel. It's called a smart komatik. A komatik is a sled, so they can carry it along behind their snow machine, and it'll tell how thick the sea ice is at any particular point and things like that. Then they can report that back to the community, and the community can have near real-time information about safe areas to travel, unsafe areas to travel, conditions of sea ice, and that sort of thing. So those are just a few examples of some of the kinds of research. I love doing what I do because I get to play around with all kinds of research in all different areas, and I really enjoy that, but especially I get to work with a lot of really awesome young folks. So I won't belabor this one. Just to give you an idea, there's still a lot to do and a lot going on. We'll be in the Kluane region of the Yukon, working with First Nations for the first time coming up soon, and a number of other projects that we have going on. So I'm going to leave it there. And with that, say thank you very much. And if there are any questions, I'm happy to take them. Oh, yeah? Yeah.
We had 6 Inuit that were involved in the research, and they were all male. I was wondering if there any organization does anything specific to try to get Inuit women involved.
So if you didn't hear, he said he just came back from Pond Inlet doing some field work, and there were 6 Inuit that were working with them. They were all male. And so the question was whether in our program we're doing anything to encourage or recruit women into this process as well. And yeah, in fact, what we have found is it's at least 50%. In some communities it's 60 or 70% female working on these projects. It varies from community to community, but we've had actually really good luck recruiting both men and women, and we have research mentors who are probably the majority have been women, which also helps, I think, to provide that kind of role model. Yeah. Yeah.
You were talking about identifying invasive species. If you do get invasive species in seawater in the Arctic,
Yeah, so the point was made that it's would be pretty difficult, to come up with something that you could actually do about invasive species if you do find them in the Arctic. And you're absolutely right. The reality is, if they're there and getting established, it can be really difficult. I mean, we've seen around the world how difficult it can be to deal with invasive species. But catching them early is important, and that's really the goal here. But yeah. Well, there's. Yeah, so you can. There are certain protocols you can establish, and some of them are in place. There's questions about whether all the ships are necessarily following them in terms of where you're allowed to dump your ballast water and these kinds of things. But Kim Howland, who works with the youth on this project, is also doing studies of ballast water to understand what's in that water to start with, so that they can establish, cleansing protocols and that sort of thing too. So absolutely, trying to stop it before it happens is the best approach. Yeah. Okay. Oh, do I have time for one more?
Quick one.
Okay.
If there's an issue that comes out from the results of the stuff done, you're going to do something like help them alleviate the situation, or?
Okay, so if there's something that's found to be a problem, let's say they're monitoring the river and there's issues that they find. Yes, what we generally do is we work with the youth in the community to find the funding to follow up and follow on with whatever that work is. So we part of that sort of capacity building among the youth in the community is helping them to develop the skills they need to go out and find the funding, and so we work with them for that, as long as the community and the youth are interested. Yeah. All right, well, thank you very much.
Yeah, thank you, Eric, for such a fascinating talk. And we have 2 more talks left. How to conduct research on ocean microplastics in 4 easy steps, and the not so glamorous life of a marine biologist, 10 reasons why saving whales is tough work. But before that, we're going to have a short 10-minute break, so you can maybe get yourself another drink, and I'm not sure there's too much food left, but you can maybe scrounge the last morsels. Yep. How's that you going to do?
Other way.
Okay.
Sorry.
I don't tell you about. Oh, oops. Whoops. What am I looking for? I can mute it out, I think. Yeah.
I just was wondering. I have one video.
Okay. That should work. Which is your this not so glamorous? And did you embed it? Or is the video just in here somewhere?
Both, I think.
Okay.
I don't know.
Alright. Hang on just a second. More complicated than you think.
Oh, I know.
Look at the remote.
This one? Oh, no.
No, the remote for this big TV that this is. I brought two. Well, you gotta be like right in the corner.
I guess because it's like...
The Vancouver Aquarium. Okay, that's better. Muted. Okay. Oh, okay. Was that it? Okay. Holy slides.
I love slides. This one.
Oh. This one? This one. You just gotta hit play, right? Where was the slide? It's this one, right? You probably know better than me. There's like a play on when you click on the slide, right? Yeah. There you go. Yes. No, automatically. Maybe because we weren't in slideshow mode. Right? So if we go from the current slide to... There we go. Okay. 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 Go all the way to the beginning. We'll minimize it, but we'll eject your stick. So I put it right here. I'll just copy it to that folder to make people feel good. No problem. That's what I do. And this one of them was Ruth's, right? Yeah, that's it. Okay, I'm going back to input. Wait, during the day? Yeah. Stuff that's kind of been playing.
They never allowed me to put a notification on.
Yeah, what's the deal with that? What? Who's they? I don't.
These are for the speakers, but nobody's used them yet, so you might as well use them. Save you a trap. There you go. Oh wow, yeah, ridiculous. But also ridiculous too. Just even more ridiculous as to what's happening right now. Where do they think all these fires and floods are happening? Like, it's no longer just happening, you know, in the Arctic. Vancouver was inundated with smoke. But the problem, do you know, actually, I think one of the largest problems is people try to confuse just in general, they want people confused because people are, yeah, no, you're right, terrible. Yeah, so bad. Thank you for showing that to me.
This one, which one?
Right in the middle. Nope. Presentation Microplastics.
I think the problem is we're all scientists, so we named it really sensibly exactly the same thing.
Yeah.
All our talks the same.
Before I changed it, it was an acronym of the title, just a series of letters.
Excellent.
How to study microplastics in 4 easy steps. I don't even remember. It looked like somebody just smashed their hands on the keyboard.
Okay, we're going to continue with our next talk. So our next talk is Stephen Chastain. He's going to talking about his research in microplastics. He's currently a researcher here with Ocean Wise's Ocean Pollution Research Program, and he works primarily on analytical techniques for studying microplastics in ocean water. So, welcome, Stephen.
this have a pointer? I think it does.
It did work, but I got it on the floor, so I don't know if it
Ooh,
still works.
nope, still works. There you go. Ooh. But not on the screen. Moot point. Hi, everybody. I'm Steven, as she introduced me. And I'm studying microplastics in ocean water, specifically the Arctic Ocean water. I'll be getting into how that happens shortly. But just to make sure we're all on the same page, raise your hand if you've heard about microplastics in the news lately. That is almost everybody. So I can give you a quick primer on what they are. This doesn't have its own slide, but instead I can share this super cool artwork that's behind my huge title here. This is a kind of a joke parody of a famous Japanese painting, The Great Wave off of Kanagawa, except if you look closely, the entire thing is made out of tiny little microplastic bits. I did not make this. I'm not nearly talented enough at art. But this is by Bonnie Monteleone, I believe. That's the attribution I saw the most via Pinterest. Super cool. If I got that wrong, I'm sorry, YouTube. I didn't mean to. That's just the name I saw, so I tried. So microplastics are anything in the ocean that is less than 5 millimeters long and is made of plastic. It doesn't matter if it was made that way on purpose or if it got that small by breaking up into little bits over time. But the problems they present are quite substantial now that we've begun to realize what we're up against here. They've been known to be in the ocean since the 1970s, but it's only been in probably about the last 15 years that the research really took off, because several studies found that the concentrations are going like this over time, like exponentially increasing. And on top of that, the amount of plastic that we're producing for our use on land is also increasing exponentially, along with population and wealth around the world and things like that. So this is one of those situations that we need to get under control quickly, because in addition to the studies finding just how many there are out there, there's been a lot of work done on health effects of both marine animals and, well, I guess only marine animals so far, at least as the ocean is concerned, and more importantly, their ability to work their way up through the food chain. So microplastics are particularly dangerous in the ocean because there's a lot of critters that eat stuff that's about the same size as microplastics. Just these guys over here, these Arctic char, would probably be prime candidates to accidentally gobble up little tiny pieces of plastic if they were maybe embedded in something else they wanted to eat, or tangled up in zooplankton or something small that they find delicious. So as those eat it, then maybe something else bigger might come along, like a trout or something, and eat them. And when it eats him or her or however, then it will eat every little bit of plastic stuck inside that fish, and now it's stuck inside the next fish, and so on up the food chain. This raises the question of human health impacts, which I will not get into, but there is some evidence that it may be getting into our food through that way, mostly through shellfish, because we don't usually eat the stomachs and gastrointestinal tracts of things that we fish, which is good. And shellfish being the exception, we eat the whole animal. You take the mussel, you scoop the whole thing out, or the oyster, whatever it is. And those guys are the worst for that because they'll eat anything that floats by. They just put their little feelers out and go like this and grab particles. And they don't care what they're made of. And plastic, we believe, smells a little bit like oil or fat, which means it's delicious because everything that's fatty is really delicious. So they'll eat them and then they'll just get lodged inside until something else comes along. But that's the importance. Here's the how. This is what we do in the lab and in the field, and how we come up with those estimates that tell you there are X thousand microplastics per cubic meter in Canada Place's seawater. We found about 1,300 per cubic meter of seawater. So if you imagine like a large crate, it's a meter in each direction. That's not a meter, but you get the idea. It's a decent number, and it's biologically relevant for little critters like zooplankton on up the food chain. So, step one, you need to go out and get them. There are several ways to do this, and it depends on what you're studying. If you're studying them in fish, you can just go out and catch some fish. And as they say on Mythbusters, the difference between messing around and science is how much of it you write down in your notebook. So in that case, you can just be keeping some notes about where you find your fish, how big they are, what kind of species they are, and then you'll know what they might have eaten. But I focus mostly on water. That was the initial project I was hired to help with, and I'm sticking around now that I've finished my, my internship with the aquarium to help us refine the way we do this. Right there, you're looking at a manta net. It's named that because it's got little wings, I guess, like a manta ray. I guess that's how that name comes around. You will tow this off the side of a ship as you're going around whichever ocean you're sampling. And you want to get that nice and far away from the ship so that the bow going up and down doesn't create a lot of turbulence in the water. Ideally, you want it to look like this when you're sampling, nice and calm, so that you can know exactly how big that little rectangular opening is. And then as you tow it for a certain distance, you can do some geometry and figure out X centimeters wide by Y centimeters into the water times Z, however many kilometers you pulled it along, equals the volume of microplastics or the volume of water that you sampled for microplastics. So once you've done that, you pick up your net, bring it back up onto the ship, turn it upside down, wash it from the outside in into a bucket, and then you can sieve that to whichever size you want through like a little shaker thing, and then you have your sample. We actually do it a bit differently than that. It's similar concept. I don't have a photo of it because I just don't. But we use the seawater loop in the bottom of our a couple different vessels that help us with sampling. We have one that actually Eric just went out. Hi, Eric. Thank you. And helped us to collect just this last summer from the Arctic. They will take a pump that's about 5 meters under the ocean surface, right at the bottom of the hull, and it goes through an initial, I think it's 1 centimeter, it's a really coarse mesh, and it goes through that, and then it will come out again in basically I think it's just a sink tap, right? Yeah, it's like a normal tap that samples ocean water, and we turn that on, and we just spray it down for a certain amount of time, and we know the flow rate of that water, and then we'll put that right through our sieve. And that saves us the issue of having this out in the air where stuff might blow away, or stuff that that didn't actually come from the ocean might stick to it. So we do it that way to keep to eliminate the potential for contamination as we're sampling. Number 2, we go back to the lab. That is my coworker, Stephanie. She is, I'll get onto the yellow outfit thing in a second. That's the next slide. But we got to get the water and the microplastics out of the water. There's a couple different ways you can do that. We have one. I'm skipping to the second bullet here first. Our lab's creation was to take advantage of the fact that plastic is water repellent. Remember I said it tastes like oil and might smell like fatty food if you're a sea critter? That's because it's actually made of oil, ultimately. Plastics are made by originally from petrochemical byproducts, but now we do it on purpose. But the stock for most plastic polymers is oil-based or petroleum-based. So they retain that repellence of water as they become plastics that oil has. And we take canola oil, very high-tech lab reagent. We buy it from I think Shoppers Drug Mart or somewhere like that. And through a couple different trial and errors, we came up with a method to mix the oil around with the microplastics in a jar of water, or actually in a funnel like that she's holding right there, which looks super cool and like the stereotypical chemistry stuff that Ruth was talking about. The white guy scientist with a beard, I don't know any of them. So we separate it out through there, and if you look closely at the bottom is a vent, or not a vent, what's that? Spout. So we can turn a little knob, drain the water out the bottom. After a while, the water goes down, the oil's on top, and then we stop right before the oil drains, and that's where all of our plastic got trapped because it's been coated and it doesn't want to hang out with the water, it wants to hang out with the oil. And that's been how we've been doing them so far for the Arctic. There's a couple other ways you can go about this. The density-based, that's my first bullet here, was developed. It's older. It's an older method. I don't exactly remember who came up with it first, but props to them because it's pretty creative too. The densities of plastic are pretty close to seawater, which is annoying because you have to sample all different depths to make sure you're getting all different types of plastic. Pulling that net along gets you what's floating, but some other stuff is neutrally buoyant, likes to hang out a couple meters below the surface. And then there's other types of plastic that goes straight down to the bottom. I'm thinking PVC that piping is made of specifically is a lot denser than seawater. But most of it's pretty close. And there's a couple different ways that you can go and get some extremely expensive fancy salt that has tungsten, sodium polytungstate in it. And I think it's like $800 for 100 milliliters of that stuff. It's insane. But you can use that to, it's very heavy. So when you pour your microplastic sample through that with the sodium polytungstate in it, everything that is seawater level but solid will float on top of it if you leave it for it's like 48 hours. That's one of the reasons that we're not crazy about that method. Also, the $800 per 100 milliliters is a little absurd. But fortunately, there are other options like our oil method. And then a lot of times when you're going through and separating from your sample, there's a lot of other gunk in the water. There's a big issue that I've run into with some water samples called transparent exopolymer fibers, which sounds kind of sci-fi, but all we're really talking about are some little precursors, apparently, of sugar-like molecules that grow in strings out of algae in the water. And that's how they chain together. If you see algae under the microscope, it's like a bunch of little beads, kind of. They put these out first, and then the cells grow out and catch up to it. They clog up my stuff all the time. And the easiest way to get rid of it, if I have to, it's not, well, not all the time, but maybe half of the samples I get have some of this stuff in it. We can just stick it in some hydrogen peroxide or potassium hydroxide. Hydrogen peroxide being the exact same stuff you use to disinfect a little cut. It bubbles and it will slowly but surely break that stuff down. And potassium hydroxide is a basic solution that, for whatever reason, dissolves a lot of stuff but never harms any of the types of plastic that we find. We've tested it on quite a few, and that's kind of cool. It's lucky, because acids, that's not the case. Acids breaks the plastic down, and then you did all that for nothing. So, bases, acids, in this case. Next, once you've got all that stuff filtered out onto a nice flat Petri dish with a white background that's nice and brightly lit and easy to see, then you get to do what I spent 4 or 5 hours today doing, counting microplastic particles and trying to figure out from looking at them what's plastic and what's not. It's fun. I'll start you on this upper left one right here. The blue thing is the plastic, if you couldn't tell. Right there. This is actually a little bullseye target-like design on our Petri dishes that helps us find things when we have to take a photo and then go back to it. But that's what we're looking at there, and I think that was polyethylene, if I remember right. So that's the same stuff that drink bottles are made of. And it was just a little fragment. Our software makes the world's smallest font on the scale bar, but I want to say it's probably about 1/5 of a millimeter long. So we usually measure these in micrometers, so that's about 200 micrometers. That's small for a fragment, but now we're going to go to the bottom left there. You see some fibers. That red one right here is, I think that was polyester, if I recall correctly, and it is about 14 or 16, somewhere in that range, micrometers wide. And for comparison, human hair is about 40 to 70 micrometers wide. So this is about a half to a third the width of a human hair. If you run your finger along a dusty surface of furniture in your house and you pick it up and look at it, if you look extremely closely, about the smallest speck that you can resolve with the naked eye, if you have good eyesight, is about the size of that fiber. And coincidentally, it's also probably the same stuff as that fiber. A lot of household dust is microplastic fibers from fireproof carpets and from your clothing as you use it. And the right side, it's blue, obviously. It was transparent, and I don't remember what it was made of, but it looks cool, and there it is. Above it, I think, is a contamination fiber. It's either contaminant or something natural that looks kind of like a microplastic fiber but isn't. And I think that's the same case with these other fibers in the lower left there. The one I was interested in that photo is the red one. We've got a series of criteria that we've developed in-house through trial and error to find out what a plastic is made of from looking at it. They work okay for the most part. If we're wrong a lot, so is everybody else. I'm not ashamed to say that. So we want to keep track of our success rate in identifying microplastics visually, and we can. I'll just go to the next slide. 3 slides ahead, and then I'll go back to those 2. Don't worry. So a big problem. I should back up a bit. A big problem with researching microplastics is you can't always tell from looking what it's made of. And there was a study from Korea that we love to talk about when people say, why are you wrong all the time? That found that the average misidentification rate of a microscale particle like that when you're looking for plastic is 70%. So we are a little low, a lot lower than that, haha. And if we're still not sure what a particle is made of, we can use the FTIR spectrometer, which is the Fourier transform infrared spectrometer. That is a scanning device that uses infrared light, which you can see on the spectrum right there, is between about 1/10 of a centimeter and I guess 10 or 100 nanometers. That's 1/10 of a micrometer. A nanometer is 1/1000th of a meter. A micrometer is 1/1,000,000th of a meter. And that is just where we are looking at our particles. So fortunately, they're still big enough that they interact with infrared radiation. And when you shoot any kind of electromagnetic light or radiation at something, it reflects back an extremely specific amount, or extremely specific spectrum on the bottom there of which wavelengths come back at you and which ones it absorbs. Every substance in the universe has a unique fingerprint of this, and that is the key to figuring out what those little particles are made of. This one at the bottom here, you can't see the text, but this is from a real sample that I put up here as an example. The orange is the ideal spectrum for what we think this one was. So when we scan it, we get the black line. And then expert interpretation, not me, I don't know what this is doing here except for a little bit, is needed to determine how it matches up to a spectrum that somebody else already figured out some other time. We have a library of about 30,000 of them. It's called the Know-It-All Library, which I think is just perfect for what we're doing here. And what we are looking at is a few specific peaks, because you're going to look at this and say, what the heck? That's not the same thing. This is doing all sorts of stuff here that the orange one isn't, and yet the machine said the black and the orange match. But what we really care about is this here, and that right there. And the rest of it is interference from stuff that grows on the fiber or whatever that was, I don't even remember, that grows on the particle while it's in seawater. So I know that this wiggly stuff on the left is carbon dioxide, so this is a ridiculously sensitive instrument. It's got 2 crystals that come together like this with the particle in the middle, and it's still reading a few molecules of carbon dioxide from the air that were somehow in between the two. Like we're all holding our breath and it still comes through. No. So the rest of that on the right there, I'm told is proteins and peptidoglycans and other things like that, that is what you get when you point this thing at something that's alive, basically. If you pointed it at a person, you'd probably get protein from the keratin in our hair or something in our skin. And bacterial cell walls are made of peptidoglycan, and we get a lot of stuff from, what's it called? Cellulose, that's it. Cellulose from algae that grows on the outsides of these particles. And that's actually pretty important for understanding where microplastics go in the ocean, because you're going to look at this particles like this. That red one is what we're focusing on. That's my plastic. The rest are little bits of zooplankton, I think, some unlucky fellows that got sucked through the intake. Sorry, fellas. The red right there, it looks clean. It looks like it's maybe just come off of the aquarium vests right now, but it's actually almost always going to be coated in biofouling like that. So it's going to have plant life, it's going to have bacteria on it, it'll have little fungi on it, and it actually, it's a little microscopic ecosystem growing on the outside of that. And as it gets denser, as more stuff attaches to this thing that used to be just able to float up and down pretty easily, it's going to gradually sink through the water column. And the latest knowledge out there in the public right now is we think that most of the plastic that's ever been in the ocean has actually already sunk to the bottom. And they've even found microplastics at the bottoms of trenches. I'm not sure if they've sampled Marianas Trench yet for microplastics in the sediment, but I know they have sampled some trenches in the Mediterranean and around Africa that had microplastics in them, and they also found it at the bottom of the Ross Sea in Antarctica. So they are everywhere. Let me go back to this. Speaking of fibers that you find that are not microplastics from the ocean, a big hiccup in research for the past 5 years has been keeping contamination out of the samples. I said that household dust is made out of microplastics a lot of the time, and lab hold dust is also sometimes made out of microplastics. This device right here that this faceless fellow here is working in is a laminar flow hood. So there is a filter, there's like a HEPA air filter in the top there where that dial is, and that's where the intake of the air goes through. That'll filter the air down to 1 micrometer. So that's enough filtration that we're pretty confident nothing that we don't need is getting through. And then the air will go down towards the sample and then back out towards you. That's good as long as you're not working with anything that smells bad or is dangerous. So we do our safe work in there. The rest of the time, we need to be extremely careful to keep the thing covered whenever we're moving around, and we need to keep our clothes covered with something that doesn't shed fibers, which is what Stephanie's wearing right there. We call them the banana suits. That is made of impermeable plastic sheeting, which is not very comfortable, but it does keep your clothes fibers. Like if I did that, I probably just made 60,000 of them come off. I don't know. But when you start looking for this stuff, you find it everywhere. We aim to be state-of-the-art with our contamination controls, so we are making very sure to do all of our work in there if we can, covered up if we can, and if we can't do any of that, then we'll put out laboratory blanks or procedural blanks, and we'll run as if there was a sample and do our extraction method, but only with deionized filtered water, and then we'll find out if there's any particles in that, then we know they came from us and not from the ocean. And if they look similar to that, we'll go back into the other samples and say, what's this one doing there? That's not supposed to be there because it's the same as the procedural blank. It's never going to be perfect, but we can get pretty darn close. So a lot of the work that I do is involved in figuring that out. That's pretty much all I have for the structured portion of our presentation, but I'm happy to take questions now. Right here in the front.
You found that there are microplastic deniers, sort of in the same way that there's climate change deniers?
So the question was, are there microplastics deniers in the same way that there are climate change deniers? No, not that I've heard of. Not yet. Not yet. I'm sure it'll happen eventually, but we're ready for them. We have photographs. Right there? Oh, good question. This one always comes up. The question was, what are my thoughts on degradable plastics? I'll preface this by saying it's my personal opinion. We don't have an official position because we're still looking into all the different variations. Compostable plastics and biodegradable plastics and bio-based plastics are 3 different things. Sometimes you can have all 3. Sometimes you can have a combination of them. Bio-based, I'll start there, means that it's made from a derivative of some sort of agricultural excess or waste. I think they usually use sugarcane stalks or corn husks or something like that. And that, I'm not sure, is always biodegradable, and I'm not sure is compostable. Biodegradable specifically in this case means if it's out in the environment, like let's say I have a biodegradable cup, I just drank all of my beer out of it and I just chuck it in the ocean. What's going to happen to it? Don't do that, by the way. I'm not condoning that. If it's biodegradable, it will completely chemically break down in the ocean into whatever it's going to turn into. We call it mineralizing. That's a less fun way of saying you can burn it and it will turn into carbon and oxygen, like completely into new substances. This is where I'm going with that. The problem with biodegradable plastics is sometimes they're manufactured as a blend of a biodegradable and a non-biodegradable polymer. And what you get then, plastics are made out of chains, and the chains are put together into sheets. So if you have a sheet, what they'll do is they'll take a sheet of polymer that's degradable and then a non-degradable one, and they can like alternate them or something. And they say, this is a 50% biodegradable plastic. And the problem is, once the biodegradable parts are gone, it's even more quickly becoming microplastics that are non-biodegradable. And then lastly, compostable. Those are, to my understanding as of right now, I was just looking at this a lot this week, they are only compostable right now in an industrial facility or a centralized municipal waste plant because they require an enzyme whose name escapes me, which sounds pretty cool, but they require that in a concentration that can only be achieved in a bioreactor. It does exist out in the ocean, in the environment, but never enough of it. So if you took a compostable fork and chucked that in the ocean, don't do it, it might as well not be compostable at all. But if you put it in the compost, like you should, then it will most likely somehow end up being soil. And we're looking into that one right now. Yeah. Oh, yeah.
I know this might be completely off the topic, but is there any, is there still radioactive pollution coming from Fukushima?
Ooh. We had a study on that, I think. Not us. So the question was, is there still radioactive debris coming from Fukushima? I believe that someone other than me researched that at OPRP, the Pollution Research Program, last year. And if the report isn't finished yet, it will be soon. From what I remember, the short answer is no. And if there was, it wasn't ever going to be dangerous in BC. It dissipated quickly enough that it wasn't going to be a concern here. So, yay, good news.
And I got just a little plug, if you are interested in biodegradable plastics, the lecture next week is by a chemical engineer who studies and manufactures, I think she primarily just studies potential biodegradable plastics and how they work. So if you want to know a lot more about that, come along to the lecture next week. So our final talk is by Brittany Vizona, and she's a researcher here with Ocean Wise's Marine Mammal Research Program, and she's going to tell us about what it's like to be a whale researcher. So, welcome, Brittany. Thanks. You know where it is? I should have.
Okay. Thank you. Yes, I'm too short for this. Oops. There we go. There we go. Okay. Well, good evening, everyone. I'm going to talk a little bit about the glamorous or not so glamorous life about being a marine biologist. So the kind of 10 reasons why saving whales is actually quite tough work. So most of us have got into the marine environment at a young age. I know myself, I wanted to be a marine biologist when I was 2 years old, and that's what I told my parents. And it was by these things of why I wanted to. And now I'm working as a research biologist with Ocean Wise's Marine Mammal Research Program, which is really cool, as they've been doing killer whale research for over 30 years. So it's the longest running continuous study on killer whales in the world, which is pretty exciting. They've done all sorts of different things, like looking at photo identification. So this is used for population modeling, looking at births and deaths. We've also looked at a lot of acoustics, so between populations and within populations, differences in dialects, looking at genetic diversity between populations and how killer whales pick their mates. Our most recent research works with drones, and we use them to measure body condition and health of killer whales. We also have other researchers that work on beluga whales, so looking at contact calls between mothers and calves and how that's affected by underwater noise. We have one researcher that's working on pinnipeds, so seals and sea lions, looking at their populations and their diets. We have a North Coast office in Prince Rupert that's looking at humpback whale research. They are doing a photo ID catalog, seeing humpbacks that are actually wintering in the area instead of going south. And they also do harbor porpoise surveys up there to look at habitat use and how that's affected by anthropogenic sources. We also have the longest running citizen science program, the BC Cetacean Sightings Network. So this is a network that collects sightings of cetaceans, whales, dolphins, and porpoises, as well as sea turtles. They're kind of the honorary cetacean. And we get volunteers, so anyone from commercial vessel operators, recreational vessels, anyone from land that sees a whale, dolphin, porpoise, or turtle to report it to us. We have quite a large database, and we use that for many different research projects. So although our job may sound quite glamorous, like we're running around in boats all day, taking photos, it's not always like that. There is some not so glamorous aspects of the job, a lot of paper writing, a lot of grant writing, a lot of staring in front of computers, which involves us eating a lot of chocolate to get through the day. So I'm going to talk about the top 10 reasons why saving whales is actually quite tough work. Reason number 1, finding funding. They never tell you this, that before you can actually do research, you actually have to go and find the funds for it. So most of our programs, you have to go and find the funds for. Our BC Cetacean Sightings Network is all funded by the Government of Canada's Habitat Stewardship Program. OceanWise does fund some of our research, and sometimes we have to be a little creative of how we find our funding sources. One example is the Wild Killer Whale Adoption Program. So through symbolic adoptions of killer whales, all of that money goes towards research. So a lot of our genetics work as well as our field work gets funded through that. So if you want to know more about that program, go to killerwhale.org. Reason number 2, data management. So you always have the hopeful goal that you'll be out in the field and that's your whole job as a marine biologist. It's all a lie. It's maybe 5 to 10% of the time, if you're lucky. Most of the time, you're sitting in front of a computer, which isn't all bad. There's a lot of data analysis to be done, especially after field season. For other programs, it's pretty much always sitting in front of a computer, such as the Sightings Network. So we get all sorts of sightings in from many different platforms, so these all have to be verified. We have to send emails to follow up with observers and thank them, as well as putting all of those sightings into a database, and then you also have to do analysis on it all. So it ends up being a fair amount of work, but you do end up with pretty maps at the end. So we do get a lot of sightings from that network in particular, and then we can do a lot of different research with it. So a lot of our conservation-based research is based on sightings network data. So we can use this data to help us look at the abundance, occurrence, and distribution of cetaceans along our coastline. So this can help with figuring out areas that could be designated as critical habitat and that are of high conservation concern. And this is quite important because in BC, we do have 12 out of 23 species of cetaceans, as well as all 4 species of sea turtles that are in BC waters that are at risk under Canada's Species at Risk Act. So it is quite important research. Reason number 3, braving the elements. Even if you're not working in the Arctic, it is still very, very chilly in BC when you're out there, whether you are on a boat or on land. So some of the vessel-based research that we do is we do killer whale, humpback whale photo identification. So with killer whales, you ID them by taking photos of their dorsal fin on the left and right hand side. So that patch there, that kind of light gray patch behind the dorsal fin, is called their saddle patch. So it's different for every single whale. It's like their fingerprint. So that's how we ID out killer whales. For humpback whales, you have to get a bottom photo of the tail fluke, and we can identify individuals from there. So then we can monitor birth death rates and see if the population is increasing or decreasing. So that's some of our field work. Other field work that we do is our photogrammetry study. So this is one of the projects that I'm involved with. So we take drones, we basically fly them over killer whales and take aerial images of them. And then from that, we can take measurements, and we can compare those measurements of how big these whales are in relation to prey, and then compare that also between years and within seasons. So we can see if they're getting enough, for example, for resident killer whales that just feed on fish, we can see if they are getting enough food after the major salmon runs and how they're doing after the winter season, and then between years as well. So with the drone there that we launched from the vessel, that's the Scanna, our main research vessel, we can determine if there's healthy whales based on how round they are, basically. We can determine if they are pregnant, so if they're really round around the midsection. We can also look at pregnancy loss rates. If we see a pregnant female one year and then the next year we don't actually see that she has a calf with us, as pregnancy loss rates are quite high in killer whales. We can also see if individuals are quite thin, which means that they are not getting enough food or there could be something wrong with them. But if they are very thin, exhibiting peanut head syndrome, which you can see right there around the eye patches, that it's quite narrow, that means that they're not getting enough food. So that's the main point of this project, is to look at their body condition in relation to their food source. We also have land-based field research. So we have up in Prince Rupert, we have a harbor porpoise study that's looking at habitat use of harbor porpoises in relation to vessel disturbance and vessel noise. And we're doing a pilot study in Saturn Islands. We started that this summer and we'll hopefully continue that on as well. But all in all, these tend to be long days. Even if you're in the lab, as well as in the field, you can have quite long days, anywhere from on average 6 to over 12 hours. Sometimes you're on a vessel for days, weeks. Sometimes you don't see land for a while, which leads into reason 5, seasickness. You unfortunately never figure out if you're going to be seasick until it's too late. It's always a good time, and let me tell you, it's not fun if you are. Reason 6, dead things. We deal with a lot more dead things than you would think. In order to monitor cetacean populations, we need to know what threats may be impeding the recovery. So whenever you do have a whale that washes up on shore, you have to figure out the reasons why. So this involves doing a necropsy to figuring out why. You can either be on one side of the scale where you really enjoy it, or on the other side of the scale where you don't enjoy it so much. It kind of depends on how you feel about cutting into an animal and how you feel about smells. Usually it's not good. And sometimes you get tasked with not so great samplings. So for Carla here, she was asked to collect a fecal sample of this whale. So it's a very dirty job. But from this, we can identify things like causes of death, whether it's based on a ship strike. Fair amount of cetaceans off of our coast get struck by ships. Some of them are non-fatal, other ones are fatal. Other things that could be the cause of death are ingesting too much marine debris, as well as a buildup of pollution in their system, so toxins, or being caught unintentionally by fishing gear. Reason 7, wild animals are sometimes very uncooperative. So they frequently become tangled in fishing gear and marine debris, and it is very difficult to untangle them. So you can only imagine trying to untangle a humpback whale. So you have almost a 50-foot animal. You're trying to get up close to them. They're fairly erratic in behavior when they are entangled, and then you're trying to untangle them with a knife basically on a very long pole. So it's very difficult, which is why only the professionals do it. But if you do see an entangled, distressed, or a dead marine mammal, please call the 1-800 hotline. Reason 8, staring at spectrograms. So you think that listening to cetaceans is really cool, it's a great job. What they don't tell you is that you're going to be staring at black and white screens for hours on end after the field season. So this is a spectrogram, which is basically frequencies of sound over time. So you can look at call rates, you can look at contact calls between mothers and calves, and looking at when they are more active in their vocalizations and echolocation. And that can be related back to underwater noise. As we are having more and more vessel traffic, that means more underwater noise. And so this causes more stress to the whales, which may prevent them from going to foraging areas or going to areas that they use for socialization or breeding. So this is important for us to look at how noise affects cetaceans. And sometimes you have to collect some dirty samples. So reason 9 is whale snot. Yes, we have collected whale snot. So using a drone, we can fly it through the blow of humpback whales, and then we can collect a snot sample. From this sample, we can look at the microorganisms that are in the whales. And so once we've established kind of a baseline for healthy individuals, we can compare that when there's any changes, so we can see when there's pathogens that are present. And another dirty reason of the job, number 10, poop samples. We do collect lots of fecal samples. We have one master student that's collecting killer whale fecal samples at the moment and looking at hormone levels in the samples, so seeing how stressed out populations are between southern residents and northern resident killer whales. There's a lot of other things that you can find in fecal samples. You can look at genetics, so which individuals they're coming from, and we can also look at diet as well. So we have one of our researchers that works on pinnipeds, so you can see the pinniped sample there on land, and from there you can collect things like bones and do genetics to figure out what their diet is as well. So there's a lot of messy reasons and tough reasons of being a marine biologist, but there's also a lot of really great ones. Yeah, whales are awesome, so you kind of suck up the rest of it because they are amazing. But even if you didn't pursue marine biology as a career, you can help out. And so there's multiple ways that you can do so. One is choosing sustainable seafood. Picking seafood that's from a sustainable source. You can dispose of your waste responsibly, so less pollution that's in the ocean. Always reduce, reuse, recycle to help reduce entanglements, get rid of any gear that's around or on the beaches. You can help out with a shoreline cleanup as well. Or you can help with research by adopting a killer whale. And if you are on the water, always be whale-wise, so make sure that if you're in with 400 meters of a whale, that you slow down to 7 knots. You never approach them from behind or from the front, you always approach from the side, and you always stay 200 meters away from killer whales, as it is the new regulations. If you do see a whale, dolphin, or a porpoise or sea turtle, please report it to us. You can be another data point on our map. So we have many ways to report. We have our 1-800 line, we have an email, we have the website, logbooks, and our whale report app, which is the best. So it's free. It's available on iTunes and the Google Play Store, and it has a lot of really cool features, so you can figure out which whale you are based on photos as well as descriptions. Once you do report your sighting, you get a pin on the map, so you can always see all the different places where you've seen cetaceans along our coast, and you can go back and read those reports, which is really cool. But the best part is that all the sightings that come in real time from the Whale Report app are going to be sent directly to the Whale Report Alert System, which is a brand new application that is going to be launched in the next couple of weeks. So this is a partnership that we have with the Port of Vancouver and Port of Prince Rupert. And the point of this is it's going to take those real-time cetacean sightings, send them to commercial vessels, and it's going to alert them that whales are in the area so that they can take mitigation measures to reduce ship strike as well as reduce underwater noise and their physical disturbance to the animals. But RAS is not available to the public. It's only available by invitation, and that's because it's going to pilots and captains of large vessels, so they get the alerts so that they can reduce their measures, and then they can choose if they are going to take action to avoid whales if safety is permitting. So basically how it works is you spot a whale, you report it in real time to the whale report app, it gets validated by us and sent out to these mariners, and then they can choose what they're going to do, whether they're going to slow down, alter course, keep in mind that there's whales coming up, or if they're just going to dismiss the report altogether if it's not safe. And you can help out by reporting your sightings in real time and help out with this initiative that we're working on. So all in all, it's a really great job, even though there's some dirty parts to it, and it's tough work, but you can never say that working with whales is bad. So thank you for listening, and any questions? It is late, so.
Yes. Okay, well, thank you so much for such a fascinating talk. Absolutely brilliant. And thank you all for coming. I hope you had a brilliant night. And I hope to see you here for some other events in the future. So, thank you.